So good morning, everybody. I hope you're still awake. Indeed, it was a very intensive 2 days, but very interesting too. So bear with me for a while, and I hope you find interesting what I'm trying to say. So today I would like to discuss, rather than present with you, how a private company could go from research-oriented towards project-oriented, and why this is also necessary for us to do so. So we would like to say that we convert scientific knowledge into applications, user-friendly decision support software. Okay, some of you have seen this slide yesterday. As I explained yesterday, there is a global trend of increasing emerging infectious diseases, of which vector-borne diseases, which means diseases transmitted by, for example, insects or arthropod species, is increasing not only in the developing world around the tropical belt, but also in developed countries or countries at higher latitudes or in the south. So it's not just something that you will encounter when you go traveling. It is something that is existing right here, right now, and increasing. There are several drivers of change. Some of them are global, some of them are local. Global trends obviously are increased mobility of people and increased mobility of goods. We also know that climate change is a fact. I think it's quite safe to say that around this audience. So that's another thing that will contribute to this increasing trend of emerging infectious diseases. Let's be clear, it's not the only reason. Then, of course, there's also local changes in which land use and land cover changes are one of them. Societal changes, people going more for recreation into nature reserves and encounter these insects or arthropods more regularly. And environmental awareness, which might lead to the increase in abundance of intermediate hosts, for example, roe deer or bank voles or mice, for example. So that's another contributing factor. So these drivers of global change, we can sort of split it up in two groups. First of all, we can consider the pests or the pathogens, which will be, the pests, so the insects, which are traveling around the globe in containers. And you all know that this is a huge network of boats and ships going around the globe. As you can see here on this picture, these are major sea routes. So you can see, if it's red, it's high density. So you can see between, for example, Europe and the US, and then down to the African belt, and then from here to India. There's an increasing amount of mobility, and things get quite rapidly from one side of the planet to the other side of the planet. The same holds for the people. This is a schematic representation of the density of air travel by people. So again, you can see the high density between, for example, the Americas and Europe, and from Europe back to Asia Pacific and Asia itself. So we could say that the pests are traveling around the world with the goods, and the pathogens are traveling around the world in the people. Once these pests reach a certain target area, then the fact that they establish themselves there depends on climate, distribution limits and survival. The number of generations is all affected by these parameters. So in fact, for vector-borne diseases, today is the ideal world. They have everything they need to go around the globe and to establish itself somewhere else. So the day before yesterday, we were talking about visions and missions. We have our own vision and mission at the company. And our vision is to bring spatial tools to the heart of the decision making, so that people that are in the field or have to make the decisions are able to do so in a very informed way. And so our mission, therefore, to achieve this is to develop user-friendly spatial decision support systems that will help them to do that. And we would like to say that we convert data, of which nowadays is plenty around, into information. You can find many different data sources across the internet. Some of them are reliable, some are not, some are pre-processed, some are not. So getting those together and in an integrated way present them to the decision maker is what we do. So our products therefore will generate you high quality data and save you time and reduce costs. So what do we offer? When we started, and I will explain the history in the next slides, we were acting more like a research department or a research group. We would offer contract research and consulting activities. Consulting activities, for example, to the pharmaceutical world, where they would have a specific question, or, for example, the European Center for Disease Prevention and Control. Since about 8 years, we are also developing products. The main reason for which is, as you all know, when you are participating in the research project, money doesn't come in at a steady flow. It comes at quite broad intervals, half yearly if you're lucky, yearly if you're not so lucky, or even longer when you're very unlucky. But we at the company have a policy that we don't hire people on a project basis. We hire people for their expertise. We want to invest in that expertise, so we want to keep these people with us beyond project durations. So that means we have to pay salaries every month to everybody that works with us. So having this sort of ad hoc and very irregular system of payment is not very good from that perspective. So we had to find other ways to generate income and develop products that will be a recurrent income and will enable us to be more flexible in terms of salary payment. So who are we? This is Guy. He's the CEO. He started the company in 2001. I joined quite rapidly. We started working together in 2003, and then I officially joined the company early 2005. And so in total we are now with 14 people, 8 different nationalities, which is quite extraordinary for a small company like ours, but we're very proud of this diversity. Also good gender balance, as you can see from the pictures, also something we are quite proud of. Most of our activities are in Europe, or Europe funding. Some of it is quite limited now, is overseas, and that's mainly in Guadeloupe in South Africa. We have a permanent representation, and other activities in Africa, but it's lower than it used to be. So there's a clear focus on vector-borne diseases, and we cannot work without international networks. We have been participating in a lot of research projects, especially in the vector-borne community, which is how we met Professor Petric some time ago already. So these international networks are crucial for our development and for our information. So how did we develop? This is a quite complicated schematics just to show how we like to combine projects and like to develop clusters of projects, because then we can cross-fertilize between them and capitalize on knowledge that we've gained from one project into the next one. So it all started with Guy's PhD, and then in the next slides I will briefly discuss. And the second seed project was Modris, which was the inventory of mosquitoes in Belgium, and I will explain that too. These two, together with the FP6 projects, Eden and FP7, Eden Next, and the founding project from the European Center for Disease Control, all led to a development of software with the support of the European Space Agency called FECMA. And that's how we started developing our products. So basically all this here is either contract research or research, and that all led into this final, let's say, pillar of our company, the product development. So let's go back in history, and we are now the 1990s, and Guy is, or even the end of the 1980s, and Guy is now traveling to Africa. He graduated as a vet master, and then he went to Rwanda first for a training in the veterinary clinic, then moved to Tanzania, and finally ended up in West Africa, in Togo, which is where we are now. So in Togo, you have a vector, this is a tsetse fly, Glossina palpalis, which is transmitting sleeping sickness. A lot of you may have heard of sleeping sickness for humans. It's also a problem for cattle, which not many people know. In fact, in cattle, it reduces the milk production, and people's livelihood depend on this, so it's quite a serious problem. So the Food Agricultural Organization wanted to prioritize areas where you had to treat against sleeping sickness. So what he did, and he was one of the first, with the supervision of David Rogers, professor at University of Oxford, to use satellite imagery to both map the species, the vector, and the host. Because the cattle had different breeds, and the local breed was very low in production of milk, but quite resilient against the pathogen, whereas imported cattle was very susceptible to the disease but had higher milk production. So we had to sort of combine the spatial distribution of the vector together with the spatial distribution of the host to be able to prioritize the areas where you had to combat the disease in the first priority. He then continued using the satellite data to try to understand the ecological factors that would drive both the presence of the vector and also the pathogen, and was then able to optimize the sampling strategy based on that. So this was the first work that was done. This was using satellite imagery already in the early 1980s, which was quite early. This was using NOAA data, so quite coarse resolution at that time, still about 10 kilometers. By 10 kilometers still, in that period, this was very advanced work. So let's fast forward now to Belgium. And this is work that we have been doing about, I'd say, a little bit more than 10 years ago. So everybody's talking about invasive species and invasive mosquitoes and how this is a threat already 10 years ago. The problem is, in many countries, we don't know which mosquitoes are there, which ones are present. Even native mosquitoes is outdated or not even ever done. So the last inventory for mosquitoes in Belgium was done in the 1960s. So when you're talking about change and climate change and emerging diseases and you don't know what your baseline is, then what are you talking about? So what we did was do an inventory of mosquito species in Belgium. And for that we developed the different stratifications, urban, rural and natural, because obviously some mosquitoes are more linked to humans, others are more linked to wildlife, others are more linked to livestock, and we wanted to capture the full spectrum of all the mosquitoes. Then we had 3 teams in place that would go out and sample in a lot of the spots, and to determine the sample size, we started playing around with the variability that we would observe in the landscape to find the optimal number of sample size that would be required to end up with spatial distribution models like this, where red is a high probability of finding the species and blue would be a low probability of finding the species. Let's fast forward. Well, this is sort of the same time. We were talking about invasive mosquitoes. And this little female here is well known by some of us. This is Aedes albopictus. This is a mosquito which in 2008 was only observed in Italy. This was the first work that was done under the flag of the European Center for Disease Control, where Dr. Francis Schaffner, Ducheyne, and other people, and Stian, they went to Italy and they went checking in every municipality almost, where can we find this mosquito? Because it was a serious problem. It's not only a vector of, for example, chikungunya, dengue, and Zika, potentially, but also gives you huge allergic reactions when it bites you. So it's just a very annoying mosquito. And in 2007, which was the trigger for this exercise, there was a local outbreak of chikungunya in Ravenna, in Italy, which was something unheard of before. So there was a clear need to know where can we find the mosquito, where is it now? So based on the information we got from this, we started developing models. This was the model that was based on the current suitability, and then we started doing a multi-criteria decision analysis to establish where this potential spread could be of the mosquito. So this map, keep it in mind, because this is where it is now, 10 years later. So if you go back, it's a pity that they're not on the same slide actually, but we managed to find that it would potentially go up to Paris and in the Netherlands, and that's exactly where it is today. So clearly there is a way of combining satellite information and environmental information to be able to say something about the spatial distribution of potential vectors of disease. So in the meantime, the situation got even worse. It's not only that the vector was spreading, also the diseases were more recurrent. So I was talking about the first outbreak in Ravenna in 2007 of chikungunya, which was the first case. It was actually the proof that the pathogen travels with the host and that a vector is already established without any further control. So this person came from Pakistan, I believe. He was already ill with chikungunya. He came into Italy. He was bitten by Aedes albopictus, and then there was a local transmission of chikungunya around his area where he lived. And so in total, I think about 200 cases of chikungunya emerged in Italy. So it was a big reason for concern. Since then, we have been observing this pattern of local transmission of dengue and of chikungunya in Italy, in France, and it's coming regularly now. So again, there's a big reason for concern. So, what is the problem? We need to know where this vector is, clearly. There is a lot of reasons why we want to know this. But as you all know, going to the field takes time, takes a lot of money, and is very labor intensive. So we have to find ways to optimize this work. And the cost of these field surveys can be reduced by combining strategic sampling and using as a back reference spatial models that are based, for example, on remote sensing data. But that also requires a lot of expertise, because not everybody is proficient in remote sensing. Certainly not the decision makers. They don't know how to process remote sensing data from scratch up to something that will give you a risk or a probability of occurrence. So how can the satellite imagery then contribute? Well, I'm sure I don't have to explain a lot how satellites work in this audience, but you have polar orbiting satellites. They will measure the environment. You will have, for example, NDVI or whatever other vegetation index. You will also have temperature. And in our specific case, we are looking at a coarser resolution, about 1 kilometer, which is okay for a continental model or a countrywide model, with a high temporal resolution, so that we could get daily measurements. So naturally, we would go to, for example, Moody's data or Sentinel data, which will have these characteristics. And so we will measure at some of these grid cells the presence or absence of the mosquito, or even abundance. We can color them, so let's assume that red is present and green is absent. And then we will combine this with our satellite information data, use either a statistical function, using generalized linear modeling, for example, or zero count models if we're looking for abundance, or even machine learning techniques like random forest or boosted regression trees to go from where we have the gaps to a map that will give you probability of occurrence. And this map is the maps that we've established in Moderisk and that we now routinely produce. We went even further and wanted to see how we can optimize the sample size, so we invented a virtual vector. We had our own truth, our own reference data set. We made a probability map and then back sampled from there to start playing with the sample size. So this is our the ground truth of our virtual vector. And then we started playing around. We were also playing around with fuzzy functions, etc., to come up with a presence-absence model. But in general, for example, what we saw is that you need balanced data sets. So you can't have an overpowering of the presence or the absence of a certain category, because then it's very easy for a spatial distribution model to predict, for example, the absences, but you're interested in the presences. Imagine if you have 900 sites which are absent and 100 which are present. If it just classifies everything as absent, well, you've had 90% correctly classified, but that's not what you want to know. So you have to balance your data sets. That's one of the things that we established, and also we established sort of the minimum sample size you would need to be able to come up with a realistic model. How do we differ then from a research group? Because until now, a lot of the things that I've been saying is research. I agree. Well, we go further. We go from the research program, for which I'm responsible in the company, to a feasibility and demonstration stage, and really interacting with the end users to be able to assess their needs. It's something that was also mentioned by Antonia several times. You need to engage with the end users because you have to contribute to the societal needs. You can invent technology, but if nobody's using them, well, what's the point? And for us as a company, this is a commercial value. We will not invest in technology if nobody will buy it. It's kind of stupid to do so. So we have to go from this R&D program to product. And believe me, this takes a lot of time. The development for VecMap took us, say, roughly 10 years. That's a lot of time to invest in your own company. And it's only possible through supporting funding programs like the ones we had for, from ESA, for example, where there's 50% co-funding, so that means you still have to invest your own 50%. That's a lot of money. So how did we go from the idea to the software? So you can define different technological readiness levels. So let's say at level 1, it's just an ID, and you have to go up to level 9, which is when it's commercially available and ready. Fully documented, you have your salespeople, you have your marketing people, and this last step takes time and money. We are finally there, but it took a lot of time. So very, very briefly, what is the main product? It's a software which is integrated. It gives you a mobile app to go on the field to do field sampling. It gives you a GIS system that will permit you first to plan the sampling campaigns and then follow up on them. So everything that is synced from the mobile app to the server is immediately visible on the GIS system. You have a web administrator function that will set up the whole environment for your specific workspace, and you can enter all the lab results that are found on the ground. So it's a fully integrated system. We call it a one-stop shop for everything that you need for this kind of exercise. You could find bits and pieces on the internet, no problem. A lot of researchers have their own specific tool, but making them work together, that's the big challenge. We now want to go one step further. So until now, we have been focusing on, say, country-wide, continental-wide models. We want to go from area-wide to urban environment. We want to go from vectors of disease to urban pests, like bedbugs, for example, and from the outdoors to the indoors. To do that, we have obtained a grant from the H2020 program, specifically to boost your position in economy. It's a specific grant for small and medium enterprises. A very competitive environment, only 5% chance to get the grant, so it's really hard work. And we got it in the second stage. So now we have the resources to make the final step and to boost the software on the market. So what value do we offer to our clients? We would like to believe, or we are pretty sure, that we can help research institute and public health decision makers at reducing their service cost by at least 60% and significantly increase the data quality and data sharing. The worst thing that can happen for a research institute is that you have a PhD student, PhD is finished, data, if you're lucky, is on a hard disk. If you're not lucky, it's lost. And you have to start from scratch again for the next PhD. So having this in a streamlined and harmonized data set will permit you to capitalize on previous data that was collected. So you can focus on the research instead of the technology, and the public health decision makers can improve their decision making with smaller teams, not smaller people. For the pest control, we are still looking at the unique value proposition, but we believe that we can increase the efficiency of doing the pest control in an urban environment by 2, and the same for private pest control companies that will go to, for example, hotels to treat against pest control, increase their revenue and increase their market share. Very important to note is that with this pest control, we also want to enable the market to move from carpet bombing, so using very toxic products, to a more integrated pest management where we'll use our knowledge of the biological cycle of, for example, the mosquito to act earlier in the cycle and prevent what we call the vector or the bug to be a pest. That's how we like to call it. One final ongoing example. This is an exercise we have been doing for the World Health Organization, where we were mapping the Aedes albopictus and Aedes aegypti in the Eastern Mediterranean region, which is a very broad region going from Morocco all the way to Pakistan, and where we have simulated the current suitability for the mosquitoes and then the potential future distribution. So we used a lot of our resources, or data resources that we had available. We also looked at entry points into different countries to be able to do this exercise. So all of this is impossible without a great team and without great people, and I cannot stress enough how great this team is. Thank you very much.